Okay, I'm back. We had just left off introducing transcription. And again, this is section 10.2 of modern biology. Steps of transcription. RNA polymers is when it comes together. The strands bind. Helicase is when it separates the two strands. Polymers is when it comes together. RNA polymers, the primary transcription enzyme, synthesizes RNA copies of specific sequences of DNA. Ooh, that is a loaded sentence. RNA polymers initiates RNA transcription by binding to specific regions of DNA called promoters. That's an important vocab. Promoters. The promoter marks the beginning of the DNA chain that will be transcribed. In eukaryotes, promoter marks the beginning of a single gene. But in prokaryotes, a promoter may mark the beginning of several functionally related genes. So, eukaryotes are the more advanced, and a promoter marks only one gene in a eukaryote. But in a prokaryote, which are the more simple cells, it can mark several genes. So, when RNA polymers binds to a promoter, the DNA molecule in that region separates. Only one of the separated DNA chains, called the template, which I was talking about earlier, is used for transcription. RNA polymers attaches to the first DNA nucleotide of the template chain. Then it begins adding complementary RNA nucleotides to the newly forming RNA molecule. In figure 10.7, oh, here it is. Not really sure what this is showing. It's showing the binding of the nucleotides. Where was I? Okay. Notice that the complementary base pairing determines the nucleotide sequence of the RNA chain in transcription, just as it does in DNA replication. The base pairing rules are identical to those in DNA replication, except that uracil pairs with adenine. So, everything in RNA is the same as in DNA, except for uracil takes the place of thymine. So, uracil pairs with adenine instead of thymine pairing with adenine. Transcription continues one nucleotide at a time until the RNA polymers reaches a DNA region called the termination signal. So the promoters are they're kind of a signal that transcription should start, and the termination signal is the signal that the transcription should stop. So you could kind of think of it like this, like the promoters are promoting that transcription comes there. So it's like, hey, I'm promoting, come here. And termination is pretty easy. You're terminating the transcription. So the termination signal is a specific sequence of nucleotides that mark the end of a gene in eukaryotes and may mark the end of several functionally related genes in prokaryotes. We already talked about the relationship in prokaryotes and eukaryotes. At the termination signal, RNA polymers releases both the DNA molecule and the newly formed RNA molecule. All three types of RNA molecules are transcribed in this process. So all of the messenger RNA, transfer RNA, and ribosomal RNA are all transcribed during this part. Products of transcription. The products of transcription, called transcripts, pretty easy to remember, are the different types of RNA molecules, including mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, which is ribosomal RNA. Although the instructions for making a protein are copied from DNA into mRNA, all three types of RNA are involved in the synthesis of proteins. Following transcription, mRNA moves through the cells of the pores of the nuclear membrane into the cytosol of the cell, where it will direct the synthesis of proteins. So, all the forms of RNA are being transcribed, but they're also synthesizing these proteins. So, they're being synthesized in some kind of form, but as soon as they're synthesized, they're going on to synthesize proteins. So, messenger RNA, which we talked about, takes the information from the nucleus and it takes it outside of the nuclear membrane out into the cytosol, and it helps like give out, I guess, essentially the blueprints of the synthesis of proteins. So, yeah. Again, I haven't read this before, but we've been talking a little bit about it in class. So, if I don't have the right idea, please leave a comment. This is just what I'm trying to come up with, if that makes sense. So, yeah. That, I believe, is the end of chapter 10.2, and you can see for my new video of 10.3, because I'm probably going to record it right after this, since my test is today, since it's 12 o'clock. Thank you for watching.